welcome back to session 5. Good afternoon. My name is Holger Hein. I'm your host for session 5. I'm dialing in from Spain. It's already dark here. You can see by the bad light source in my back. I would definitely prefer being in Brazil right now, but that's what it is. Together with Orit, who talked this morning, I'm co-chairing the Standards and Technology Working Group, and I can only repeat her words that you should want to feel free to reach out, whatever problem you have for experimental design techniques, reach out to us, and we hopefully can help improving your experimental design and make your data quality as good as it can be. So today it's an honor for me to chair session 5, the last session of the day. Thanks for hanging in, but it's definitely worth it because we have 4 exciting talks in this session. As it is already tradition for the HCA conferences, we're on a very tight schedule. But that's therefore some housekeeping at the beginning. So we have 4 talks. You can again ask your questions in the chat. And we have a Q&A session right after the talks. You can ask your questions in English, in Spanish, or Espanol. So, cualquier pregunta, ponedlas en el chat y vamos a traducir todo. I will do my very best, at least. I will have some help from John and Julio to translate whatever comes in Spanish or Portuguese. So, without further delay and to keep the schedule, I would say we start right away with the first talk by Silvia Sookoian. And it's about from cell to tissue to complex diseases, perspectives from a physician. Christine, please.
Hello, everyone from Buenos Aires, Argentina. I am delighted to share with you this talk, and I'll also thank the conference organizers for the kind invitation to address this fantastic topic. What if I told you that single-cell research may help to understand the pathogenesis of complex diseases? I will share with you 2 clinical cases through which I will address the physician view of the power of single-cell research to understand the human health and disease. This is a case of a 53-year-old male with non-alcoholic fatty liver disease, arterial hypertension, morbid obesity, and type 2 diabetes. Liver biopsy showed non-alcoholic steatohepatitis, which is the severe histological and clinical form of the disease. Since the age of 48, at the time of the first clinical consultation, patient gain weight and present persistently elevated aminotransferase levels. Second biopsy, 5 years apart from the initial one, showed the patient developed NASH cirrhosis. Non-alcoholic fatty liver disease is a condition characterized by abnormal fat accumulation in the liver, which can present hepatocyte injury and chronic damage, such as those that characterize non-alcoholic steatohepatitis, also known as NASH. The disease can progress into severe clinical forms, including cirrhosis and hepatocellular carcinoma. And the disease pathogenesis involves a myriad of factors, including genetic susceptibility and predisposing metabolic comorbidities. And despite 2 decades of extensive research in the field of NASH, there is currently no approved therapy for the disease. And diverse factors explain the low success rate in NASH drug discovery, including insufficient understanding of cellular function and patterns of gene expression. To move forward with this, let's review some key concepts of a liver anatomical structure. The liver consists of repeated anatomical units termed liver lobules. These lobules are hexagonal-shaped columns. Blood enters the lobules from the corners, termed the portal tract, and flows radially inward towards a draining central vein through the sinusoidal blood vessel. It is known that many liver functions are non-uniformly across the lobules, a phenomenon termed zonation. More importantly, liver zonation is associated with diverse liver pathologies. For example, pericentral predominance is associated with fatty liver disease and alcoholic liver disease. Here I show you that fatty liver progress to a severe disease in a zonated manner. Periportal predominance is associated, for instance, with autoimmune hepatitis. And zonation of liver expression is a key feature of other metabolic organs such as the intestine and the kidney. Then, single-cell technologies may allow the reconstruction of zonation patterns for the global hepatocyte transcriptome. Using single-cell RNA-seq, McParland and co-workers studied the hepatocyte heterogeneity of human liver. Here we can see the proportion of cells that contribute to each cluster in the liver and the physiological conditions. For example, the author found 6 distinct hepatocyte populations comprising cluster 1, 3, 5, 6, 14, and 15 that were, as expected, enriched of albumin expression. Pathway analysis suggests that there are specific functions for each of the mentioned clusters according to the periportal or pericentral zones. For example, cluster 6 seems enriched for expression of genes involved in lipid and cholesterol synthesis, and cluster 6 seems involved in cellular pathways of immune cell activation. The summary map of human liver from the work of Sonia McParland shows again zonation in the hepatic acinus. Within the acinus are hepatocytes, you can see those in red, and non-parenchymal cells, for example, cholangiocytes in green, endothelial cells, Kupffer cells, hepatic stellate cells, and liver-resident and infiltrated lymphocytes. Of note, the authors also show that liver immune cells are distributed in specific zonated patterns, and non-parenchymal cells are not merely bystander cells. In fact, macrophages are important regulators of liver homeostasis and injury. For example, in the liver of our patient with NAFLD, we can see how infiltrating immune cells attack the fat droplets. And sometimes the inflammatory infiltrate is severe in terms of amount of infiltrated lymphocytes. Therefore, tissue-resident macrophages are regarded as the immunological sentinels of the liver. As mentioned before, our patient developed NASH cirrhosis, which is a serious liver disease. Here we can see large scars in the liver, our patient, and we actually need to reveal what is kind of cells lead to these scars. Hepatic stellate cells are major contributors of the myoblast pool. Nevertheless, these cells are not the only ones contributing to liver fibrosis. In fact, macrophages, endothelial cells, and mesenchymal cells are also involved in the processes of fibrogenesis. How then these cells of the fibrotic niche interact with each other? Single-cell transcriptomic profiling of scar-associated macrophages, endothelial cells, and mesenchymal cells show multiple ligand-receptor interactions. For example, macrophages express ligands that can promote mesenchymal activation. For example, TGF-beta1. Similarly, endothelial cells can promote mesenchymal collagen production. For example, Notch3. And more importantly, interactome modeling of ligand-receptor pairs between subpopulation of scar-associated macrophages, endothelial, and mesenchymal cells in liver fibrosis provides a fantastic molecular framework for the therapeutic targeting of key pathogenic cell populations. This landmark study used single-cell transcriptomics to assess the non-parenchymal cell subpopulation in the fibrotic niche of patients with cirrhosis, and the author identified a scar-associated cluster of macrophages trend to positive CD9 positive subpopulation that expands in liver fibrosis. Using single-cell RNA sequencing analysis, here the author also showed the reprogramming of liver non-parenchymal cells in mouse liver, and they identified 2 distinct subpopulations of cells characterized by low or high expression of TREM2. And this trend to macrophages derived almost exclusively from the NASH livers. In this study, single-cell RNA-seq was used to explore the hepatic mesenchyme in the fibrotic mouse niche, and the author identified 3 distinct populations of mesenchymal cells that present zonation across the hepatic lobule. And the author found central vein-associated hepatic stellate cells as dominant collagen production cells during injury-induced fibrosis. But more importantly, the author identified a lysophosphatidic acid receptor as a therapeutic target and demonstrated that pharmacological antagonism of this receptor inhibits liver fibrosis. Let's move to the second case presentation. This is a 61-year-old male patient with also non-alcoholic fatty liver disease, mild arterial hypertension that present at the ward with fever, dry cough, headache, and mild dyspnea. And TC scan showed bilateral lung infiltrates. The nasopharyngeal swab was positive for the new coronavirus, and the diagnosis was confirmed as COVID-19. Among other laboratory features, our patient present mild elevation of aminotransferase levels, about 2 times the upper normal limit. And what could be the possible explanation of that? First, patients with chronic liver disease may be more vulnerable to the clinical consequences of COVID-19, including oxygen desaturation and hypoxemia due to severe pneumonia or the cytokine storm. Second, liver biochemistry abnormalities could be the consequence of drug toxicity. And there is a third potential but poorly explored clinical scenario, which is the possibility that the novel coronavirus may directly or indirectly cause liver injury. So here is the academic debate. SARS-CoV-2 may cause liver injury. These are molecular observations that could be in favor or against this theory. Some experts say that the low or lack of expression of ACE2 in hepatocytes explain that the novel coronavirus does not actually cause viral hepatitis. On the contrary, other experts believe that the level of ACE2 in cholangiocyte is similar to the alveolar cells of the lungs, indicating that the liver could be a potential target of the SARS-CoV-2. Let's see whether single-cell transcriptomic analysis help to understand this issue. We examined the liver expression profiling of host receptor implicated in a SARS-CoV-2 infection across the liver cell type from the McFarland database. And you focus on ACE2, TREM2, and furin. And the analysis showed that 3 human host receptors are expressed in the liver tissue. However, expression level extensively vary across the cell types. ACE2 expressed the highest expression levels in cholangiocytes, followed by hepatocytes, and the other 2 receptors shows actually levels across all cell types, from hepatocytes to all cell population. But other authors claim that the pattern of expression of this receptor is not uniform across the different cell databases. For example, cholangiocytes are the most the ones that express higher levels of ACE2 in all databases, but ACE2 expression in hepatocytes show large differences among the different studies. In summary, application of single-cell research is in the path of precision medicine. But single-cell transcriptomic is not the only one analysis we can use. In fact, and you can appreciate here that there are complementary molecular profiling technologies, for example, single-cell epigenomic and proteomic profiling, and single-cell metabolomic profiling. So although the costs have reduced, it remains an expensive approach for some Latin American countries. So collaboration is crucial to decrease the inequities in research and care of human disease. And open access of the large databases is also important. Thank you very much.
Thanks, Silvia. Now we have Silvia in the live chat for the Q&A. Welcome. Let me see. Maybe I will start with some questions. So the liver is a very highly zonated tissue, right? And you were mentioning many single cell studies, and with a single cell, we basically take their cells out of their tissue, bring them in solutions, we lose the spatial organization. So if you like think of an ideal scenario, would you rather go for the spatial, keeping the spatial context and let's say, so you would lose single cell resolution, but you gain spatial context. That would help you understanding better the function of that tissue and in disease?
Oh, thank you. This is a great question. Actually, the work that has been done in the human cell atlas is fantastic regarding the liver, because we know, realize how is distributed the population of parenchymal and non-parenchymal cells. But in the context of diseases, I think that we do need to keep the spatial context, because zonation is key in all liver pathologies, particularly in metabolic diseases. This is very, very well demonstrated, not only at the transcriptomic level, but also at the epigenomic level, that zonation is the key and perhaps the answer to the pathogenesis of metabolic diseases.
Okay, great. And then related to the diseases, we sometimes have problems to get cells out of the tissue. And you've seen a lot of pictures with different degrees of disease stage. Do you think that there can be biases when taking cells out, because some tissues might be more dense? So we might expect like a systematic bias when sampling from later disease stages.
Definitely. You are in the key question that I've been. This question is being heard at least 30 years in my career when I started as a resident, and now that I am old physician, I always listen the same, because most of the picture I show you in a presentation are the result of liver biopsy. That is a percutaneous liver biopsy. When you place the needle, actually you don't know where you are going, but you can presume you are in the left, right, the lobule of the liver. But of course, zonation implies the whole organ. So this is very challenging, particularly for studying zonation and particularly for studying metabolic diseases. Even when you take a surgical biopsy, which is larger in terms of quantity of tissue compared with a needle, even when you take this surgical biopsy, you need to go to very specific lobule. Generally, you use the left one. So this is challenging for understanding how the disease progresses, because, as you may know, the vascular system in the liver also is different according to the lobule. So this is something that we need to understand better right now, because it's very challenging from a technical point of view.
Yeah, well, we have this problem in many tissues, but I believe in liver it's particularly crucial to have that in mind when doing the data interpretation. Good, then for the sake of time, thanks so much, Silvia. And we would move on to the next talk. Thanks so much.